Hello everybody, in this video lecture we will continue covering the chemical composition of the living organisms. And as always, I'm going to use PowerPoint to help myself. So this is a PowerPoint that I'm using, right? So I'll go up. So this is the chemical level of organization. And we cover most of this PowerPoint, and we stopped on organic molecules, organic compounds. So this is where we stopped. So let's go ahead and begin. So I'm gonna pick up my pointer. So organic compounds, they contain carbon. Actually, they also contain hydrogen. So organic compounds, they contain carbon and hydrogen. And of course, molecules like carbon dioxide or carbon monoxide, they are inorganic, even they do contain carbon. Organic compounds, they're unique to living systems, and they include carbohydrates, lipids, proteins, and nucleic acids. This is not all organic compounds. We have actually lots and lots of organic compounds that students study when they take organic chemistry. Those are large organic molecules that are especially important in biology. Many organic compounds are polymers. And polymers means it's a large molecule that is made from repeating building blocks. And we call these building blocks monomers. So monomers make a polymer. Monomers, your cells, right, connect those monomers together. So chemical bonds are formed between monomers. And that's how polymer is made. And this process is called chemical reaction, and specifically dehydration synthesis. So polymers are synthesized by dehydration synthesis. Synthesis means you're making something, large molecule. Dehydration means water is removed during this process. Now, your cells can also break these polymers back into monomers, and this reaction is called hydrolysis. So polymers are broken down by hydrolysis reaction. Lysis means breaking apart. Hydrolysis, you break apart and you use water to do it. So actually, that's what is shown here on this slide. And it's simple representation. You have one monomer and another monomer, and as if water is removed, so that's a dehydration, and monomers are linked together by covalent bond. So it's a dehydration synthesis. The reverse reaction would be hydrolysis. So here you have two monomers that are linked together. Water is added, and bigger molecule is broken into two smaller monomers, right? So, and of course, it's very complicated process. Sometimes to attach two monomers, we need the whole, long chain of chemical reaction and different enzymes to do it. So it's more complicated than it's shown here on the diagram. Here's a reaction when two sugar molecules, glucose and fructose, are joined together through the same dehydration synthesis, forming disaccharide called sucrose. That's your table sugar. And when you eat this table sugar, you actually can hydrolyze it. So in your GI tract, with the use of water, with the help of water, sucrose will be broken back into glucose and fructose. And glucose and fructose now called nutrients, and they can be absorbed in your bloodstream. So we're going to start with carbohydrates. Carbohydrates, the most Well-known examples include sugars and starches. Carbohydrates contains carbon, hydrogen, and oxygen. And hydrogen and oxygen in the same ratio as water. So in a molecule of carbohydrate, you have as many molecule oxygen as you have carbon. So you have carbon and oxygen in the same amount, and you have twice amount of hydrogen. Carbohydrates can be divided into three classes: monosaccharides, disaccharides, and polysaccharides. The major function of carbohydrates is energy production, specifically synthesis of ATP. So function, carbohydrates are major source of cellular fuel. And also carbohydrates are structural molecule. Your DNA and RNA has the base that is called, sugar is, base is nitrogenous base, but they made from sugar molecules, right, ribose or deoxyribose. And then nitrogenous base, phosphorus group, but large part of your DNA and RNA is actually sugar. So here's our monosaccharides. Monosaccharides are called a simple sugar, and they contain 3 to 7 carbon atoms. And again, the same ratio, CH2O N times. That means, let's say if this N is equals 5, then it will be C5H10O5. Right, and here the most important, well, some of the most important monosaccharides. I don't want to say most, really less important, because everything important in your body, even those trace elements that you have less than 0.1%, but you cannot function normally without them. So everything is important, but those are like the most abundant monosaccharides: glucose, fructose, and galactose, and they are hexose. Hexose means they have 6 carbons. They all have the same chemical formula, C6H12O6, but they have different structure, right? That we call it isomers. Isomers are molecules that have the same chemical composition, the same numbers of atoms, but different structural organization of these atoms. And pentose sugar, 5 carbons, include deoxyribose, found in DNA, ribose, found in RNA, or ATP. Right, so I don't know, let me pick up very quickly my pen. Right, so if, let's say, if I'm talking about glucose, fructose, right, if I'm talking about glucose, fructose, and galactose, right, they have 6 sugars. So their chemical formula are going to be C6H12O6, right? So I have as many carbon as oxygen, and I have twice amount of hydrogen. So they all have the same chemical formula, but different structure. If we're talking about deoxyribose and ribose, then it will be C5. C5H10O5, right? Those are pentose sugar. Okay, where I've got my pointer. Now, disaccharides. Disaccharides are double sugar. They are too large to pass through the cell membrane. That means, actually, if you eat disaccharides, and you do eat them, sucrose is your table sugar. So you put it in your coffee, or when you make your pancakes, that's what you're using, sucrose. Lactose is found in milk. We call it milk sugar. So you drink milk, you actually getting lactose. Maltose is found, for example, in beer. If you drink beer, then you get maltose. We also have maltose shakes, But those are all disaccharides. And when these molecules enter your stomach first and small intestine, they cannot be absorbed to your blood. So because they cannot pass through the membranes. So what you need to do, what we call it digestion, you need to digest sucrose into glucose and fructose. You need to digest maltose into glucose and glucose. And you need to digest lactose into glucose and galactose. And then this simple sugar can move through cell membranes and be absorbed into your bloodstream. That makes sense? That's why if we have people who are lactose intolerant, that means those people are lacking the enzyme that would break this lactose into galactose and glucose. So they drink milk, but they do not digest it. So it moves through their GI tract and it's irritated. And that's why people have cramps and gas and some other unpleasant condition if they are lactose intolerant. Right, so how those disaccharides are formed? They form by linkage of two monosaccharides, and this reaction is called dehydration synthesis. Polysaccharides. Polysaccharides are polymers of simple sugar. Example would be starch and glycogen. Humans, we do not make starch. We don't make starch. We don't store starch. This comes from plants. Glycogen is called animal starch. So plants have starch, animals have glycogen. Polysaccharides are not very soluble. So if you look over here, these green parts, they represent simple sugar, glucose. So this is example of glycogen. So you see this molecule has different branches over here, but this green stuff, that's a glucose. So you have glucose attached to another glucose, right? So glycogen is a polysaccharide formed from glucose units, right? So how do we call it? How do we call this reaction? Dehydration synthesis again. Now, can you digest polysaccharides? Depends, right? You can digest starch. You can break down glycogen. Another example of polysaccharide would be cellulose. In your diet, you call it fiber. That's the one that you cannot digest. But what does it mean, digestion of polysaccharide? That means inside your GI tract, you need to break these covalent bonds between simple sugars, and then simple sugar can be absorbed into your bloodstream. Another group of large organic molecules are lipids. Lipids contains carbon, hydrogen, and oxygen. Now look, let me go back over here to the carbohydrates. Right there. Now carbohydrates contains carbon, hydrogen, and oxygen. Lipids contains carbon, hydrogen, and oxygen. Then what is the difference? Well, in a chemical composition, the difference is that lipids have very little oxygen, less than in carbohydrates. And this is what makes them non-polar molecules, by the way. So very little oxygen. Sometimes lipids can contain phosphorus. They are insoluble in water. Lipids are not polymers. Carbohydrates are polymers. So they are made from repeating molecules of sugar. Lipids are just large organic molecules. Here's main types of lipids. Natural fats or triglycerides, or sometimes it's just called simple fat. Phospholipids, steroids, and eicosanoids. We will talk very briefly about each of these types. Triglycerides, neutral fats, solid fats, and liquid oils. So when you eat your butter, right, that's a fat. When you eating olive oil, that would be also neutral fat. Triglycerides, they are composed of 3 fatty acids bounded to glycerol molecule. And main function are energy storage, insulation, and protection. So here we can see how simple fat, so that's a molecule of simple fat over here, or neutral fat, or triglyceride. And you can see how it was formed. So glycerol, right? And glycerol is organic alcohol. And 3 fatty acid chains and fatty acid are organic acids. Now, where is it formed? It's formed inside your cells, right? Especially cells that love forming these triglycerides are adipocytes. Now, your cells using complex biochemical chain of reaction, right? Biochemical chain of reaction and different enzymes to attach this 3 fatty acid to a glycerol molecule. And when they attached, water is removed. So you can see water is removed over here. So it's again dehydration synthesis. So this is how molecule of neutral fat look like. It's called triglyceride because it has 3 fatty acid and 1 glycerol molecule. And if you look at fatty acid, you see you have carbon, hydrogen, carbon, hydrogen. These 3 dots represent that there are many of these CH2. They just skip it, right? Because this is very long tail. And then here you have oxygen. Here you have oxygen. So way less oxygen compared to carbohydrates. Now, when we're looking at this fatty acid can be saturated and unsaturated. But the word saturated means it has something, right? If you say your soil in your backyard is saturated, you mean it has lots of water, right? So what they saturated with? They saturated with hydrogen. So saturated fatty acid have the maximum amount of hydrogen that they can have. That's why they are saturated. And they have single bonds between carbon atoms, right? Single bond between carbon atoms and maximum number of hydrogen. That's why they are saturated. And those fatty acids, they are solid and usually they are animal fats. Example would be a butter. We can have oils, right, that are saturated fats. I mean, not, you wouldn't call them oils, but you do have some plant fat, like coconut oil, for example. Coconut oil that is saturated, but it didn't come from animals. So most of the saturated fatty acids come from animals, but some come from plants as well. Fatty acids can be unsaturated as well. Then they have double bonds between carbon. And because they have double bonds between carbon, they have reduced number of hydrogen atoms. And those usually plant oils, for example, olive oil. So saturated fat, they are solid at room temperature. Unsaturated fat, they are liquid at room temperature. Now, if you look over here, right, so this is unsaturated fat, and this is what we call good fat. But if you look at this fatty acid, you can see here we have double bond between carbons, and that's why these 2 carbon atoms, they missing hydrogen, right? They only have 1 hydrogen attached. Every other one, look, it has, carbon has 2 hydrogen attached. Carbon, 2 hydrogen, right? Except of this carboxyl group at the end. But because we have this double bond, we reduce the number of hydrogen. And also this acid is not straight, right? It has this kink. Saturated, that's what we call bad. This is good, this is bad. Bad fat found mostly in the animal fat, like in bacon, for example. And here you can see that every single carbon has a single bond with another carbon. So we have maximum amount of hydrogen attached. Now, what is trans fat? Well, trans fat is actually unsaturated fat, but it has been partially saturated. And trans fat is especially bad for your health. Now, this is how it looks. Compare with unsaturated. You see how hydrogen is on the same side of the carbon, and how over here hydrogen is on a different side of this double bond. So we have here double bond, and we have hydrogen on one side and hydrogen on another side. Compared to this one, we have hydrogen on the same side. That's what trans fat is. Trans means across the double bond. And now, unfortunately, these trans fat are found to elevate bad cholesterol and should be avoided as possible. And they found a lot in many processed food, or when you fry your food. So that's why they recommend to boil everything, really, instead of frying it. Another group of lipids are phospholipids. Phospholipids are modified triglycerides. So remember, in triglycerides, we have glycerol and 3 fatty acids. Here we have glycerol and 2 fatty acids, and 1 fatty acid is replaced by phosphorus group. So this is how phospholipids are represented when we want to draw their structure. They have a head and a tail. And head and tail, those regions have different properties. Heads are hydrophilic. That means they are polar, they have a charge, and they are attracted to water. Tails are hydrophobic. That means that they don't have any charge, and they repel water. And because they repel water, they tend to be facing to another fatty acid, right? So you see, this is called phospholipid bilayer. So phospholipids form this double layer that is very important in the membrane structure. The membrane of a cell is a phospholipid bilayer, and this bilayer is formed because of the different properties of the heads and the tails. Because if you look at a cell, cells are surrounded by water. Actually, animal cells must be surrounded by fluid. If animal cell is not surrounded by fluid, it will die. So all your cells need to be surrounded by fluid, or here it says just water. And there is a lot of fluid inside the cell. Cytosol is mostly water. Now, because we have water on both sides, we have those heads that facing the water because they attract water, they love water. And we have those tails pointing away from water. And that's how double layer is formed. So here you can see structure of phospholipid molecule. So here how we represent it with polar head, non-polar tail, right? But here's our glycerol, 2 fatty acid, and fatty acid can be saturated or unsaturated. And instead of 3rd fatty acid, we have this phosphorus containing group that is called polar head. So this is the head. Right, and here's we have 2 tails. Another group of lipids is steroids. Steroids are really look very different, nothing like molecules that we just described, but they are interlocking 4-ring structure. Steroids, they have 4 rings. Example of steroid would be cholesterol, vitamin D, steroid hormone, bile salts. Another group is eicosanoids. Eicosanoids have many different forms. One of them are prostaglandins. So here's a picture of prostaglandin. So you can pretty much see how it has a ring similar to steroid, and it has the fatty acids over here, right? So that's a fatty acid. This one is not, yeah. Okay, so, but you don't need to really memorize the structure of prostaglandins, but they all derive from a fatty acid in cell membranes. Now, why they are important? Look, this is your cell, right? The cell has cell membrane, and the major part of cell membrane are phospholipids. So phospholipids in cell membrane. Now, when your cell is injured, then those phospholipids are converted into prostaglandins. And these prostaglandins, they consider to be inflammatory chemicals. So they provoke inflammation, or they start inflammation, along with many other chemicals. And inflammation causes redness, worms, swelling, and pain. And inflammation is actually a very important process in tissue repair, right? So that's how prostaglandins, well, this is just describe what is their importance, right? And also what's shown over here, if you take aspirin or Advil, what they do, they block this conversion. And they call anti-inflammatory drugs because if you can stop this conversion of phospholipids into prostaglandins, then you decrease inflammation. And by the way, prostaglandins, the name came from the place where they were first found. They found them in the prostate gland. So they found them in the prostate gland and they call them prostaglandins. But now we know they can be found anywhere in your body, and they are part of a group of the chemicals that are called inflammatory chemicals. Other lipids in the body include fat-soluble vitamins, DEC. If you put A and then DEC, so it's A, E, D, and K, and lipoproteins. Lipoproteins that transport fat in the blood. Those are made from lipids and proteins. Steroids. Steroids are also, we already discussed, steroids, they have 4 rings, they're lipids, but here example giving you is calciferol. Calciferol is vitamin D. Vitamin D made from cholesterol in skin and requires sunlight, and it's secreted into bloodstream. And what vitamin D does, it actually increases absorption of calcium in small intestine. In America, vitamin D is added to milk to promote calcium absorption. And they called it vitamin D because they didn't really want to call it steroid hormone, because vitamin D is your steroid hormone. So if they write on a cart of a milk that this is milk with a steroid hormone added, nobody would buy it, because nobody want to buy milk that has steroid hormone. That's why they called it vitamin D. So this is milk with vitamin D added. Oh, that's awesome, because vitamins are good, steroid hormones are bad. That's not true. We must have steroids hormones. Vitamin D is a steroid hormone. Proteins, another big group of large organic molecules. Proteins are polymers of amino acid. They're polymers of 20 different amino acids and joined by peptide bonds. They contain carbon, hydrogen, and oxygen, the same as our lipids and carbohydrates. Proteins also contains nitrogen and sometimes sulfur and phosphorus. So here you can see different amino acid. And if you look at the amino acids, they have the same part, right, over here, right? This part and this part, this all the same. And what is different is this R group. So based on what this group is, we have 20 different amino acids. So what is the same in amino acids? This is called central carbon. This is called okay, so this is called carboxyl group, and this is called amino group, right? So it's right here. So that's amino group because it has nitrogen, and this is called carboxyl group. It has COOH. This is actually tells you this is acid. That's why they amino acid. So this group, carboxyl group, is identification of organic acid, right? So, and hydrogen. So central carbon, amino group, carboxyl or acid group, and hydrogen, that's the part that is the same for every amino acid. And R group is what is different. Now here you can see how inside your cells, two amino acids are attached by these peptide bonds, and peptide bonds are really covalent bonds. And water is released, so from two amino acids, your cells can synthesize dipeptide. And this dipeptide can be again split into two amino acids by hydrolysis. This is a very complicated process. It's called protein synthesis, and it includes DNA, RNA, ribosomes, and probably you know something about it if you took biology, but this is very complex. But when amino acids are attached, this structure that is formed called primary structure of protein. So primary structure of protein is a sequence of amino acid that forms this polypeptide chain. Now then proteins, they are modified. Actually, they are folded in a way that either they form a helix, like this spiral, and it's called alpha helix. And what holds it together in this position are hydrogen bonds. So you see this dotted lines, those are hydrogen bonds. Or they can be folded into beta sheet. So beta sheet, right, it look like this zigzags. Or it's also called beta pleated sheet. So this is called secondary structure. So secondary structure, major alpha helices or beta pleated sheet. And also you can see how these hydrogen bonds hold them together, this structure. And then protein assumes its tertiary structure. So tertiary structure is a combination of the helices. So here's the helix and the sheet together. And it can be, for example, a globular molecule over here. That's our tertiary structure of a globular molecule. And it's held together by intramolecular bonds. Some proteins can also have quaternary structure. And quaternary structure, when you have more than one polypeptide that form this protein. Example would be hemoglobin. Hemoglobin has 4 different polypeptides that form one molecule of hemoglobin. So hemoglobin has this quaternary structure. Now proteins can be divided into globular proteins. They are functional proteins. They are compact, spherical, water-soluble, and sensitive to environmental changes. They also can have specific functional regions. An example of globular proteins would be antibodies, hormones, enzymes. Or proteins can be fibrous proteins, and they are structural proteins. They are strand-like, water-insoluble, and very stable. So they're not that sensitive to a temperature change or pH change. However, they still are. An example would be keratin, elastin, collagen, and certain contractile fibers, such as actin, myosin, right? So Here's, for example, is collagen here, right? And collagen is found in your connective tissue. You will learn about it later, but this is example of structural protein. This is hemoglobin, that is example of globular or functional protein. And also here you can see how hemoglobin has a quaternary structure, so it's made from 1, 2, 3, 4 different polypeptides. Protein denature. Protein denature is a shape change and disruption of active site due to environmental changes. So when pH changes or temperature changes, then proteins can lose their structure. It can be reversible if normal conditions are restored, or it can be irreversible. So example of irreversible would be when you're cooking an egg, right? So, well, egg, this white of the egg, it has protein. So this is active protein. It has its precise structure, three-dimensional structure. When you apply heat, this structure is lost, right? And this is irreversible damage. So there is nothing we can do now to return this protein back to its original form. Or if you just warm the milk, if you don't boil it, but you warm the milk, this is example of reversible change. So here you have active protein, inactive, but then the milk cools down, it can return back to the kind of active form, but it's not going to be the same. Yeah, so you can even taste it. If you boil milk before, it's gonna be a little bit different. I'm not sure if it's because of this lactose. It might be because of lactose as well, but it's not complete restoration of the original form, but it's still reversible. Okay, now what are enzymes and why would we talk about enzymes? Because enzymes are proteins. Right now we're talking about proteins. Enzymes are biological catalysts, and what enzymes do, they speed up chemical reactions without being changed or used during this reaction. We have millions of different proteins and they're very specific. Enzymes. Every single enzyme is really very specific. And enzymes, they lower the activation energy and increase speed of reaction. So here, if you see reactants over here, for reaction to begin, we need invest some energy first. So without enzyme, larger amount of activation energy is required. When we add enzymes, that less activation energy is required. And that's why reaction happens very quick, millions of reactions per minute. So enzymes. Often enzymes are named for the reactions they catalyze and using ANDase, hydrolase, oxidase. Some functional enzyme consist of protein, it's called APA enzyme, and cofactor. And cofactor can be either some iron or a vitamin. Actually, vitamins, you would wonder, why would I need vitamins in my diet? Because vitamins are coenzymes. So what they do, they help enzymes to function. Here you can see again just representation of how enzymes work. So enzymes, they have active site, and then whatever binding to this active site is called substrate. So here substrate, for example, 2 amino acid, they attach to the active site of the enzyme, and enzyme catalyze the bond formation. Now, look, product is released, and enzyme is released, and enzyme is not consumed, and it's not changed. So there is no any conformational change in this enzyme. Now, enzymes inhibitor very quickly because they're really important. Actually, enzymes inhibitors are poisons. They are chemicals that prevent a substrate from attaching to enzyme. So if you look over here, the green is enzyme, and here's our active site and substrate gonna feed in this site, and reaction, will happen. Now, what if we have a chemical shown here in yellow and here in yellow that attaches to enzyme and prevent substrate from binding, right? If inhibitor attaches to active site, then it's called competitive inhibitor because it competes with the substrate. But sometimes inhibitor can bind to the allosteric site. Allosteric site is any other binding site other than active site. So if inhibitor binds to allosteric site, look what happened over here. The size of and shape of active site is changed, and substrate still cannot bind over here. This is called non-competitive inhibitor. Now, if you heard about cyanide, it's actually the enzyme inhibitor, and it's inhibit a special enzyme called cytochrome excitase that is important in cellular respiration. And cellular respiration is needed for ATP production, for energy production. So, people actually die very quickly from cyanide poison because if this enzyme is not functioning, cells cannot synthesize ATP. If cells cannot synthesize ATP, they start dying very quickly, especially nerve cells. Right? Another enzyme inhibitor, acetylcholine esterase, or ACase, this inhibit the enzyme that is important in your muscle contraction, and it's cause muscle paralysis. We do use enzyme inhibitors in the medicine. Let's say antibiotics. Antibiotics, they inhibit bacterial enzymes, bacteriicides. Aspirin and Advil, we just talk about it because they inhibit the enzymes that converts phospholipids into prostaglandins and decrease inflammation. Lipitor. If you have elderly people in your family and if they have high blood pressure, I almost certain at some point of their time they will prescribe Lipitor, because what Lipitor does, it inhibits the enzyme that converts fatty acid into cholesterol. And people with high blood pressure usually have high cholesterol level. That's why they take Lipitor to prevent this cholesterol synthesis. Now we're getting to the proteins. Protein hormones. Oh, hold on. Where is my proteins? Why I get straight to the hormones? I think I'm losing some of these slides over here about proteins. Okay, so this is what I need to do. I need to add some slides over here. No, I don't. I apologize. Yeah, because we already cover all these proteins. We all good. Sorry for that. Sorry for this. Yeah, I thought I need to add some extra PowerPoint slide, but we don't. Well, just making sure you guys are here with me. What are we talking about? Because it seems like I lost the track as well. We are talking about proteins, right? We talk about primary structure of proteins, right? So that's the primary structure of proteins, just a chain of amino acid. We talk about secondary structure of proteins, alpha helices and beta pleated sheets. We talk about tertiary structure, quaternary structure. We said that proteins can be globular or fibrous, And we said, well, proteins can be in nature. Then we talk about enzymes. And we talk about enzymes because enzymes are proteins, right? So, and then we talk about enzymes inhibitors. They're not proteins, but they're just important in understanding of enzyme function. And here, another example of proteins in your body are protein hormones. For example, insulin or growth hormone. What is important about protein hormones? They must be injected. Steroid hormones can be taken orally. So if you take steroid hormones, for example, birth control pills, many birth control pills contain steroid hormones, and you take them orally because they are not broken apart by digestive system. Any protein hormones will be digested. That's why, you know, insulin, people need to do injection of insulin. They cannot just have a pill. Antibodies are also proteins, and antibodies are functional proteins. What they do, they inactivate foreign agents. Foreign agents can be bacteria, for example, or some virus. So antibodies can attach to bacteria and deactivate them. Antibodies are produced by special type of white blood cells that are called B lymphocytes. Okay, and the last group of organic molecules are nucleic acids, including DNA and RNA. Those are the largest molecule in a human body, contains carbon, oxygen, hydrogen, nitrogen, and phosphorus. And building block for nucleic acids are nucleotides. Nucleotides composed of the sugar, nitrogenous base, and phosphate group. So let's look at picture first. So if you're looking over here, shown here in green, okay, let me get my pointer. Right, so here shown in green and here shown in yellow, this is two nucleotides. So they all have this sugar, and this is deoxyribose because we're talking about DNA here. And this is pentose sugar. It has this phosphate group, and it has this base, nitrogen containing base. And the same here, sugar, phosphate group, and base, right? And then these nucleotides, they connected together forming this skeleton, sugar, phosphate backbone. That's what we call it. Sugar phosphate backbone. And then it has these bases hanging on a side. And between the bases, we have hydrogen bonds, right? So let's go back. So when we're talking about DNA, we have 4 bases: adenine, guanine, cytosine, and thymine. And DNA is double-stranded helical molecule in a cell nucleus, and it provides instructions for protein synthesis. DNA also replicates before cell division, ensuring genetic continuity. Continuity, sorry. As I said, that's your DNA. That's a double helix. Double helix means double spiral structure. And what's shown here in a kind of purple color would be this part, phosphate and sugar. That's purple part, and it's called sugar phosphate backbone. And what's shown in green, yellow, blue, or red, those are nitrogenous bases. And nitrogenous bases form hydrogen bonds, and A always pairs with T and C always pairs with G, right? Those bases are complementary: adenine and thymine, and cytosine and guanine. If we're talking about RNA, we have adenine, guanine, and cytosine, but we have uracil in the place of thymine. RNA is single-stranded molecule, mostly active outside the nucleus, and 3 varieties of RNA carry out the DNA order for protein synthesis. Well, means in DNA, we have instructions how to make proteins, Now we need to carry this instruction from DNA to ribosomes, and that's our 3 RNA that help your cells to do it. Messenger RNA, transfer RNA, ribosomal RNA. And the last molecule that we're gonna discuss is ATP. ATP is energy molecule of your cells, and it's a nucleotide. Remember, your DNA and RNA, they made from nucleotides. ATP is just a nucleotide. It has a ribose sugar. It has the nitrogen containing base, specifically adenine. Remember, we have A, T, C, and G. So here we have A. So ribose and adenine called adenosine. And then we have, instead of 1 phosphate group, we have 1, 2, 3 phosphate groups. If we have 3 phosphate group, it's called ATP, adenosine triphosphate, 3 phosphate. We can have 2 groups, then we call it ADP, adenosine diphosphate. Or this molecule can have only 1 phosphate group, then it will be AMP, that will be adenosine monophosphate. And in these bonds between phosphate groups, the large amount energy is stored, especially between group 2 and group 3, second phosphate and third phosphate group. So what your cells do, they use this ATP molecule as a rechargeable battery. Like if you have rechargeable battery, you use this battery to do something, maybe to make your remote work. And then when charge is gone, you recharge it back. The same with ATP. Your cells actually break this bond, and this energy that is released, used to carry on cellular processes. And then what is left is ADP. And ADP is like a battery that doesn't have charge anymore. So what your cells do next, they attach this phosphate group to the ADP and making ATP again. So it's like you're recharging the battery. Well, how do you put this phosphate group to the molecule? How you attach it? What kind of energy you use? You use energy of your food to do it. You break down food, and the energy, that energy is used to attach third phosphate group to the second phosphate group and make ATP from ADP. Okay, let me see. I think, oh yeah, so one more slide. Function of ATP. So, so what it says over here, the terminal phosphates are enzymatically transferred to and energize other molecules. And let me get my pointer again. And those molecules then perform cellular work, life processes using the phosphate bond energy. So look, what's shown over here? Shown a protein that is embedded inside the cell membrane. And this is a transport protein. So what this protein does, it's carry molecules from outside the cell inside the cell. And this is example of active transport. So for this pump, or for this protein to work, they use ATP energy. So what ATP does, right? ATP is actually attaches to this protein, right? It attaches the phosphate group over here, right? And it's allow this protein to change its shape and do its work. Oh, actually here we carry molecules out, but it doesn't matter. We can carry molecules in and out, but if it's an active transport, we use ATP to do it. So now ATP is broken into ADP and phosphate group, right? And that was used to open this channel. Also, ATP can be broken into ADP and phosphate group by your muscles, and muscles contract. Or it can be let's say, chemical reactions that need to happen in your body, but they cannot happen without ATP, right? So this phosphate group from ATP will be transported to this molecule. Reaction happens, phosphate group is released, and ADP is released. So ATP is broken into ADP and phosphate group for some chemical work. That's functions of ATP. Okay, now this was our last slide. So this chapter was covered in 2 video lectures. It's called Chemical Level of Organization, and you have to watch video lecture part 1 and video lecture part 2. Well, and thank you for doing it, and I hope it was helpful.